Good evening, my friends, and I've been welcoming back to my channel this evening. This evening, we want to discuss a topic, and the topic now classification of organisms. Now we understand, say for science, different organisms dey, and one of the problems with science they face now how to classify or group these organisms. And science come begin realize say the best way to group these organisms now to group them based on the features or the structures where they get, where they similar. So maybe the ones we get backbone, we'll group them together. The ones we fit run, we fit walk, we group them together as animals. The ones we know they work, where they one position, we group them together as plants. So this whole method they very important, and they important because millions and billions of organisms they. So it is very necessary for science to group all these organisms into different groups so that we fit recognize these organisms, we fit study these organisms independently, and now wetin bring about classification or grouping of organisms. Now, the first man we will talk about, his name na Carus Linnaeus. Carus Linnaeus. This man na man from Sweden, na Swedish man, and him be botanist. That's na scientist where they study plants. And this man na him been design the first method. Na him group all the organisms where they live, all the living things where they for this world. Him put them into two major groups, and these two groups na plant and animals. So before the time where science just begin, now the major classification where all the scientists know be this. They look at organisms, no matter whatever the organism be or however it be like, they look at them as either a plant or an animal. And na Carlos Linnaeus na him been design this method. He designed this method around 1753 to 1758. Now that time him been write a lot of papers, write a lot of articles where he use divide all these organisms. Now, as Linnaeus been do this thing, this no be the only thing we do. Him can design another method. In design wetin we dey call taxonomic rank. And just like you dey hear am rank, it mean to put something for level, different levels dey. So in order for the man to adequately categorize or classify organisms, him come put them for different ranks. Now we get one small thing where they use take remember these ranks so that if I dey for exam or anywhere, I go dey remember am. And wetin I use remember am na King Pharaoh classes orders from General Shegun. This one I use am take remember say kingdom dey, phylum, class, order, family, genus and species. So all these different ranks na different ways wey this man use take classify organisms. So the more organisms been dey similar to each other, they go dey for these different ranks. And you fit use this acronym take remember kingdom, phylum, class, order, family, genus, and species. E dey very important to remember. Now, e dey important to know say species na the smallest level for this taxonomic hierarchy or taxonomic rank. So organisms wey been dey the same species, wetin they dey share na say they dey very similar in structure and function, and they fit interbreed, meaning say they fit come together and reproduce because they dey very similar. So for an example, all the human beings wey dey the earth now, we dey look at all of them as Homo sapiens. Dem dey together, they fit interbreed, they fit relate, they fit produce after themselves. E dey very important. Now, e dey important to also know that for microbiology, e get a level where they even lower than the species level, and that level we dey call am strain. So you get organisms where they a particular species, they still fit dey different. They fit get wetin we dey call variations. They fit vary one to another. We go call those ones strains. So many strains go dey. Under those strains, if you take all those strains together, then you reach species. And after that species, you go reach genus and then family, and they go up like that. Now, for sake of example, we just take two examples. The first example na human beings. Human beings, if I want to use that taxonomic rank, take classify human beings correctly, we go start from kingdom Animalia, phylum Chordata. Chordata means say they get backbone. And then you get class Mammalia, you get other primates, you get family Hominidae, and you get family Homo, and the species na sapiens. So Homo sapiens. If we take lion, wey dey for inside zoo or e dey for the wild, dey for jungle, e also get im own classification wey science give am. So make I just run that classification, make we take use am as example. You get kingdom Animalia, you get phylum Chordata, you get class Mammalia, you get other Carnivora. That one means say they dey chop meat, they dey chop raw meat. And then you get family Felidae, and then you get genus Panthera and the species na Leo. Now, down at the taxonomic rank, and it's important to know this rank. Every organism wey dey on earth dey divided based on this rank. Now, this no be the only thing. Another thing wey Carolus Linnaeus been design na wetin we dey call binomial nomenclature, and this means say every organism wey dey on earth, we dey use two names take represent the organism, and that organism dey get two names. Every organism get two names. So human being now, for science, the name of human being na Homo sapiens, and those names dey be Latin names. Now, e dey important to understand this name. That name na the name of the genus and the species. So the genus go dey, the species go dey. Those two names, we go dey call them together, and na the name wey every organism get for science. Now, e get as we dey take write am for science wey dey very, very important. If you no write am like that, they go mark am wrong for you. Now, wetin be the way? The way na say every scientific name, we dey put am for italics. You know wetin they dey call italics? You go bend am, the writing go bend like this. So na italics. And the first thing wey you need to know na say the genus dey start with a capital letter. So as you get Homo sapiens, Homo na the genus or general name, and e dey start with capital H. And then sapiens wey be the species name, dey start with small s. So Homo sapiens, e go start with capital H, e go start with small s, and they go italicize am. This thing dey very, very important. Scientists no dey joke with am, and your lecturers, they no go dey joke with am. If you look at lion too, Panthera Leo, that Panthera, that capital, that P for there go be capital P, and the Leo, the L go be small l, and they go italicize am. This one dey very important. Now, as science come dey grow, they come find out say this two kingdom method wey Linnaeus been design, wey be say all the organisms wey dey for the world, they put them for two different kingdoms, plant and animal. They come see say this thing no dey adequate, no dey okay, because they been realize say many organisms dey wey be say they no dey fit into any of those groups. You no fit call them plants, you no fit call them animals. So wetin we come do about these organisms? Na im scientists come dey work. E come get one man, the man na American and na biologist. Him dey work on living organisms. This man name na Robert Whittaker. Now, for 1969, Robert Whittaker come design his own method. He call am the 5 kingdom method. So he come expand the 2 kingdoms wey Carolus Linnaeus don design. He come expand them to 5 kingdoms. Now, those 5 kingdoms dey important to note. The 5 kingdoms na Monera, na Protista, fungi, plant and animal. Na the 5 kingdom method wey we dey use currently for science now. So Monera dey, Protista, fungi, plant and animal. Now, if you look through it, Monera, wetin dey for Monera? Monera na bacteria and archaea. And bacteria and archaea, they be prokaryotes. If you go check our video on prokaryotic cells, we don't discuss them for this our channel. If you know to understand wetin prokaryotic and eukaryotic cells be, if you go there, go watch that video, you will take understand better. So our bacteria na, our Monera na bacteria and archaea. Our protista na majorly amoeba and paramecium. Now them be the organisms wey dey under protista. Then we get our fungi. For our fungi, we get organisms too, such as our mushrooms, such as our yeast. All these organisms, they dey under fungi. And from the protista come up, those ones na eukaryotic cells them be. They no be prokaryotic. So now only Monera be prokaryotic. From protista to fungi, they be eukaryotic. After fungi, you can't get plant and animal. Those ones all dey together. So that na Robert Whittaker's 5 kingdom method when they use classify organisms where they live for the earth. Then lastly, you get some organisms where we know they call them organisms. They no be living organisms, but they be living they be structures, but they no be organisms. We know they call them organisms sometimes, or sometimes for science we they call them acellular entities, and these are viruses. Then they very tiny and very little, but they're important. We they also study them for science because them been they cause a lot of diseases to both man and animals and humans. So it is important to study them also. This one they very important. So this one will bring us to the end of our class. For this our class, we don't discuss classification, which means the grouping of organisms to different categories. We don't discuss wetin Carolus Linnaeus do, taxonomic rank wey in design, and binomial system of nomenclature wey in design. We can't discuss Robert Whittaker, wetin him do for science, we be say in design 5 kingdom method of classification. That one bring us to the end of our class. Thank you for listening and have a wonderful evening.